Hi everybody, thanks for coming today. I'm really excited to share with you the research that I've been working on for the past 10 weeks regarding Magnolia macrophylla and the complex relationship it shares with its floral visitors. So before I really get into the crux of the research that I've been working on, I thought it was important to sort of go over the floral biology, ecology, and morphology of macrophylla flowers. So here we have a great time-lapse taken by Dr. Juan Lasada here at the Arboretum, and it shows the 2-day protogynous flowering period of macrophylla flowers. So protogynous refers to the fact that it's first in its female stage, you can see here, referenced by this really beautiful cup shape, but as it moves into its second day, it's going to turn into its male phase. And you can see the anthers are going to dehisce there, produce a lot of pollen, and the stigmas, the female reproductive organs, are going to brown and become non-receptive. What really sets, so here's a great diagram of the macrophylla flower. So you can see an outer whorl of tepals here. So those would be sort of a proto-petal. They can act as a reward for certain pollinator species. And in the center, you can see the stamens, again, the male reproductive organs. They would produce the pollen and the carpels. So notably on the carpels, there are these sort of curved bits here. Those are what's known as the stigmas. I'll be referencing them a number of times throughout this presentation. What really sets macrophylla apart from other species of magnolia is the secretions that it outputs in its female stage. So you can see here these sort of dew-like beads that form. There's no known hypothesis on the function of these, but one idea that sort of exists is that they act as a way to sort of catch pollen from visiting pollinators. However, they happen to have a really unique effect on a few specific clades of insects, which I'll touch more on in just a moment. But first, I thought I'd introduce the natural pollinator of macrophylla trees, the beetle, as well as the fly. So these guys are totally unaffected by the secretion, and that's likely due to their coevolution with the trees. They would have been around well before macrophylla diverged and likely have either developed or already had an existing immunity to the effect of the secretion. These guys would pollinate the flowers mostly by foraging on pollen and tepals and sort of inadvertently pollinate by moving pollen from a male to a female flower as they move from flower to flower and tree to tree. But let me show you what happens when a bee, a honeybee in this case, comes into contact with a secretion. So here we have a female honeybee. She's been out foraging for the day. And you can see here she's making a lot of contact between her legs and the beads of secretion there. Now, in just a second, she's going to start to do what I call escape behavior, which only lasts for about 30 seconds, where she's going to try and make it out of the flower so she can move from flower to flower and continue foraging. Now, she's unable to, for a reason that I'll touch on later in the presentation. But for now, just understand that 30 seconds is a really short interval of time. I've seen bees trying to escape flowers from anywhere from 10 to 20 minutes, as they're, like I said, really interested in moving from flower to flower. So here you can see she's become lethargic, and she's going to stay in that position for several hours and eventually die, which is quite notable. And we think it has something to do with the secretion, primarily because we've only seen it in macrophylla trees, and we've only seen this secretion in macrophylla trees, and we saw that she touched the secretion. So there's a bit of a corollary there. And so when you have a really neat sort of natural observation like this, it's worth consulting the literature. And I was able to find this great paper by Dr. Leonard Tyne, published in 1974, where he references the flowers of M. macrophylla and M. ashii, which are two subspecies of the macrophylla family, or macrophylla species, that seem to act as a trap for certain bees and moths, but not for beetles. So again, that sort of backs up that the co-evolved pollinator of these flowers is not affected by the secretion. However, bees, like we just saw, and moths, notably insects that diverged after macrophylla trees, are affected. So where does my research fit into this? I've primarily broken it down into 3 stages: observation, chemistry, and behavioral studies. Observational studies involved me getting out in the field a lot, observing what happens when floral visitors and pollinators alike enter flowers, what they're doing inside, how they're pollinating, as well as bagging experiments where we take a frozen bee, let it wake up inside a flower, watch what happens when it comes into contact with secretion, and that's how we got that great video I just saw. And finally, collecting the pollinator and floral visitor community. So I'm an undergraduate at Dr. Lynn Adler's lab at UMass Amherst. And when I go back in the fall, I'm going to be taking a lot of the insects that I took with me, sort of figuring out what species they are, what subspecies, and building a picture of what kind of visitors are coming to these flowers and what the effect that their visits bring. We're also looking a lot at the chemistry, which involves collecting a lot of secretion and floral tissue. And that's because we have a collaborator at the Royal Botanical Gardens at Kew, and they're going to be doing LC-MS analysis, which is really similar to NMR. It's basically going to tell us what compounds are in the secretion. If we can match them to a known toxic compounds to certain insects, like especially bees, that's really interesting. We've probably figured out what's going on. But we think it might instead be an unknown neurotoxin, just due to the really short period of effect that between contact and lethargic behavior. And finally, I'll be doing behavioral studies. So that's going to involve a lot of taking secretion, applying it to species of bees. We're going to look at sweat, honey, bumble, and carpenter bees, as well as a few species of beetles. And hopefully we'll be able to observe the same observations that we saw out in the field in the lab, notably bees becoming lethargic, but beetles not so. And if we do see that, that's great, confirms our observations. But if we don't, it means that there's some other aspect of floral biology that we weren't aware of out in the field that's instead having this effect. Here's some research that I did in the past few weeks that sort of goes back to the video of the bee trying to escape the flower I mentioned. So on the left, you can see a tepal there. This would be an inner tepal, and this would be the inside where the bee was trying to escape on. I've taken sections, which you can see here increasing, from each part of the tepal. And you can see here these sort of wax crystals. So I took these images with cryo-SEM, which is a form of scanning electron microscopy. And those wax crystals indicate that this would be a really waxy surface, mostly so at the bottom. As you can see, there's sort of a decrease in density there. But again, this would sort of prevent the bee from being able to grab a hold of the tepals and prevent the bee from escaping. And it sort of references what Tyne said about the flowers acting as a trap. So finally, I think this research sort of begs the question, why deter floral visitors? Because if you have a flower and a bee is coming along, and it's not necessarily decreasing the fitness of the tree, but it's not providing pollination, it seems sort of extraneous to emit a secretion that instead kills bees. So I think one idea is that it could just be sort of an evolutionary byproduct, maybe an offshoot of some other floral biochemistry going on, or just something that got dragged along from early evolution, and maybe there was a pollinator that's now extinct that did make use of these secretions, and they just happen to kill bees now. However, I think there are a few hypotheses that exist in literature that could go as far as explaining what we're seeing in the field. The idea of microbial defense, where plants will put toxic compounds in their nectar to deter microbial action. In this case, it could be deterring microbes brought by bees to the flowers. Nectar robbery. In this case, it would probably be something along the lines of secretion robbery, where flowers are putting secretion with toxins that prevent bees from coming by, stealing that secretion, but not providing any pollination in return. And finally, the idea that I think might be closest to realistic is the idea of anti-herbivory. So often in the field, especially with carpenter bees, because of their large size, I would see stigmas with carpenter bees that it just chewed straight through them, like you can see here. And so I think if a plant wanted to deter herbivory, a great strategy would be put a toxic compound in its stigmas, and in the secretion on its stigmas that would prevent herbivory, which doesn't seem like it did in this case. Nonetheless, I'm really interested in pursuing this research as I move into Dr. Adler's lab in the fall, and I'd like to thank Dr. Adler herself, Dr. Friedman, and Amelia for really helping pull this project together. And if we have one quick question, I think we have time for that. Yeah?
Do the beetles or weevils interact with the secretion at all? Do they show any signs of anything ever?
I wish I had more observations to really tell you. I wouldn't say they go as far as to collect it, but it doesn't seem to have any direct effect on them. I don't have any interaction with it. Yeah.
All right.
You're welcome. Thank you.
Next up, we have Buster, and Buster has been working this year with Jake.
Hi, everybody. I'm Buster, and I was working with Jake this year to look into the relative contributions of climate and phylogeny to maple tree drought tolerance. So, maples inhabit a wide range across the globe. There are about 120 species in total, and that you can see they go everywhere from America all the way to China. And this same temperate band across the globe is predicted to have drier conditions in the coming decades. And so we're really not sure how this drying climate is going to affect maples, let alone other woody species. So my project this summer was measuring what's called the Turgor loss point, or the drought tolerance, in these 8 species. So the reason I chose these 8 species is to sample a wide diversity across the maple tree phylogeny. About half of these species are Asian species, the other half are American species, and about half of them are trifoliate, and the other half have simple leaves. And so the Turgor loss point is what we're really using to measure the drought tolerance. It's a great proxy for drought tolerance because, well, the trigger loss point is the point it's the water content at which the leaves start to wilt. So after the trigger loss point, the tree experiences some really catastrophic damages, including a loss of photosynthesis and a really bad xylem embolism, which could have, as I said, catastrophic damages for weeks to come. So before the trigger loss point, plants lose water exponentially. After the trigger loss point, they lose the water linearly, as you can see here. So plants that have a lower trigger loss point are more drought tolerant, and plants with a higher trigger loss point are less drought tolerant. So let's see how these different clades here, let's see what their drought tolerance is. So in the first clade here, we have Grycium and Triflorum. You can see it's pretty different drought tolerance within one clade. Macrophyllum and Mono, again, really different drought tolerance. Same thing for Davidii and Pennsylvanicum, and same thing for Negundo and Henryi. And if we mapped these characteristics onto the phylogeny, you would see a very different looking phylogeny. This indicates that the phylogeny probably doesn't determine drought tolerance much. And so this begs the question, if the phylogeny doesn't determine drought tolerance, then what kind of factors are determining drought tolerance? And so we thought that maybe something like aridity or dryness would determine drought tolerance. So here we have a graph of the median species range aridity and the trigger loss point. So species that live in drier places have lower trigger loss points, and species that live in wetter places have higher trigger loss points. And this is exactly as we would expect. What's interesting, though, is this is only the Asian species. If we added the American species to this graph, the relationship would disappear. And we're not exactly sure why this is. It's probably because American species respond differently somehow to climatic factors than Asian species. All right, thank you very much. I'd like to thank Jake Grossman, Faye, Jinhua Li for the phylogeny, the DaRin Butz Foundation, and all my other interns and colleagues here. Thank you. Yeah, I actually did. Yeah, so the technique I was using used pressure bomb, and so that, for people who don't know, and one way to get around this is using very small green branches instead of singular leaves. Also, you kind of just get used to the bubbles, and you can kind of tell even through the bubbles.
All right, thank you very much. All right, next up we have Anju. Oh, sorry, she's working with Anju.
Yes. So, hello everyone, I'm Elaine, and today I'll be talking about my project, which is finding relationship between stomata complex architecture and stomata movement. So stomata are really important to plants because they are necessary for photosynthesis, and carbon dioxide can only enter the plant through stomata. And when plants open their stomata, water vapor can also leave the plants, means that plants cannot open their stomata all the time. So stomata opening and closure are really important for plant survival. So how does a stoma open or close? So a stoma is made of two guard cells. Here are the guard cells and the adjacent epidermal cells from a leaf cross-section perspective. And our lab believes that there are two types of stomata movement mechanism. So the first type only involves the guard cells. So we refer to GC only. So when the guard cells lose water, it will close the stoma, and when the guard cells gain water, due to the decrease in diameter, the stoma will open again. And the second type of mechanism, which also involves movement of the epidermal cells, and which we refer to the GCE mechanism. So when the epidermal cells lose water, it will create space for the two guard cells to move further away from each other. And for the plants that have the GCE mechanism, their stomata can open for a short period of time when their leaves suddenly lose water. But the long-term response for water stress like this is stomata closure. So this type of response is called the runway response, but also known as transient stomata opening. So we want to know how does the epidermal cells affect the guard cells in the GCE mechanism. So I'll be looking at the plants that have the GCE mechanism and see whether their epidermal morphologies and patterning are similar to each other or not, and whether the adjacent epidermal morphologies and patterning determine how much their stomata can open. So to answer the first question, I look at the epidermal morphologies and patternings around the guard cells and group them into 4 different categories defined by Carpenter. So parasitic is basically having 2 morphologically distinct epidermal cells bracketing the guard cells. So the guard cells are being exposed to less cell wall junction, which you'll see in the other 3 groups. And so in laterocytic, stephanocytic, and pneumocytic, the middle of the guard cell are in contact with cell wall junctions in different ways. And the assumption is that plants that have the GCE mechanism tend to have the parasitic conformation because the guard cells are being exposed to less cell wall junction, which tend to be less flexible and also prevent the guard cells from moving further away from each other. So the plants that I've looked at belong to these 4 orders, and here's a list of 7 species. And previous studies have not confirmed which type of mechanism they have, but Andrew has looked into them and found runway response occurred in all of them, meaning that they all have the GCE mechanism. And now let's look at the morphology. So starting with Australia scandens, it has 3 different types of epidermal morphologies. So using the middle one as an example, this is parasitic. As you can see, 2 morphologically distinct epidermal cells on 2 sides of the guard cell. And this is stephanocytic, so morphologically distinct epidermal cells surrounding the guard cells. And here's laterocytic, 3 morphologically distinct epidermal cells on 2 sides of the guard cells. And it has a parasitic fraction of 0.2, means that 20% of the stomata I've looked at have the parasitic conformation. And next, Schisandra chinensis. It has both laterocytic down here and also parasitic, and it has a parasitic fraction of 0.275. And Nymphia thermorum, it only has pneumocytic, which means that the surrounding epidermal cells are not is not different from other epidermal cells that are not adjacent to the guard cells. So its parasitic fraction is 0. And for these 4 species, Elysium, Liriodendron, Asimna, and Calicensis, they are all dominated by the parasitic conformation. So their parasitic fraction are up above 0.9. So they all have different morphologies and patterning. So we're wondering whether these variations can cause how much their stomata can open. So to answer the second question, I compared the calculated anatomical maximum stomata conductance, GSA max, to the measured peak conductance. So GSA max is calculated by measuring the maximum pore size, the pore depth, and also the stomata density, and take into this equation. And the GS peak is being measured by cutting the leaf from the plants, allow the plants to perform runway response and further opening of their stomata. We compare the 2 data by dividing GS peak by GSA max, and if the ratio is above 1, meaning that the plant is having a higher actual peak conductance than their theoretical maximum conductance. And we found this in Schisandra, Elysium, Australia. And now we are wondering whether it's because the epidermal morphologies and patterning is causing this kind of pattern we see over here. So I then plotted the ratio against the parasitic fraction and found a p-value which is pretty high, 0.34, meaning that the parasitic conformation does not have effect on allowing the guard cells to move further away from each other. So combining our results, we know that all 7 species have the GCE mechanism. And although most of the plants that I look at have the parasitic conformation, we can now say that parasitic is a necessary trait for the GCE mechanism because for Nymphia thermorum, it only has a pneumocytic, but runway response occurred in these plants too. And also, the cell wall junction might not be a really big important factor that prevent the guard cells from moving further away from each other because in Australia and Schisandra, their parasitic fraction is around 0.2, which is pretty low, meaning that their guard cells are being exposed to more cell wall junction. However, their GS peak is actually higher than GSA max, so meaning that their guard cells move further away from each other. So for future directions, we want to compare the GS peak of plants that have the GC only mechanism to their GSA max and see whether they match or not. And also look at more species and do 3D modeling, which Andrew is already doing this, and look at how the epidermal cells affect the guard cell movement more, and for example, how the cell wall junctions affect the guard cells. And also measure more species' actual stomata conductance to modify the GSA max equation, because currently this equation does not take into consider the epidermal cells effects on the guard cells. So it will be interesting if we can modify this and also make better prediction of stomata conductance. So that's all, and I will thank you, Andrew and Missy, for their help on my research, and also Faye and all the interns for the wonderful lunch meetings, and also DaRin Butz Foundation for providing this wonderful research experience.
All right, well, thank you very much. Next up, we have Mare, and she's been working this summer with.
So my name is Mare, and I'm here to tell you that plants need nitrogen. And that's not really a surprise. So nitrogen is a key component of DNA, RNA, and proteins, and it's been shown that the way plants take up and move around nitrogen is actually impacted by carbon dioxide. So in a changing climate, we really need to understand this relationship. So to kind of introduce you to one of the main ideas that sparked interest in this project, I'm going to talk about rubisco. And rubisco is just really abundant and famous and important protein that can fix either carbon dioxide or oxygen. And which one it fixes is largely dependent on the relative concentrations of carbon dioxide to oxygen in the cell and as an extension in the atmosphere. So when rubisco fixes carbon dioxide, it leads to carboxylation, and it's this beautiful process that leads to energy for the plant. Well, conversely, sometimes rubisco fixes oxygen, and when this happens, photorespiration occurs. And photorespiration is this kind of wonky process that takes place in different parts of the cell and doesn't actually lead to a net gain in energy for plants. So people have deemed it as a really wasteful pathway, and it gets a lot of hate, it gets a really bad reputation. And so scientists have tried, or people have been trying to figure out, well, what might be a benefit of photorespiration? Because it's persisted and the vast majority of plants actually still have this pathway. And one hypothesis is that in photorespiration, it being kind of so wonky and taking place in different parts of the cell, it creates kind of like a opportunity or this excess, a byproduct of energy that plants use in nitrate assimilation. And nitrate is one of the forms of nitrogen that's most common for plants to use. So nitrogen reactions are some of the most energy intensive reactions in life. So if photorespiration is creating kind of like a boost for this, that shouldn't be looked past. So now I want to link to you photorespiration and water use efficiency. So for photorespiration to occur, like I said, you need oxygen in the cell. And stomata conductance plays a role as to how much oxygen is in the cell. And Eling did a wonderful job explaining what stomata are. They're essentially these pores that control what goes in and out of the cell. And so it's actually shown that plants that are more water use efficient oftentimes have their stomata closed more often. And this allows for less water to escape the plant, as well as this leads to higher oxygen concentrations. So I use these two lines of Arabidopsis. One was a high water use efficient line, and one was a low water use efficient line. And we saw, and it was known, that the high water use efficient line had higher concentrations of nitrogen. So we're kind of trying to piece this together and thinking, all right, so this line has more nitrogen and it also has more internal oxygen. Perhaps it's performing more photorespiration, and therefore it has more kind of like this energy boost to do its nitrogen reactions. And those are the two big main ideas leading into the project. And so now I'm talking about my project goals, which were to understand the impacts of elevated carbon dioxide on plant biomass and seed nutrition in the lines of Arabidopsis with varying levels of water use efficiency. So what happened this summer at Wells Hill? Well, manipulation happened, where lines of Arabidopsis with different levels of water use efficiency were grown, and plants were grown with varying levels of nitrogen and carbon dioxide. So you can see kind of above I had a growth chamber. Plants were either grown at 350 parts per million, which is pre-industrial revolution, or 700 parts per million of carbon dioxide, which the Earth is well on track to seeing. So next deprivation happened. Half of replicates were subject to low nitrogen nutrient solution. And termination, where 2/3 of replicates were harvested for root, rosette, and inflorescence biomass, and 1/3 of the plants are still being left to seed for mass spectrometer analysis. So my results so far have actually been pretty surprising. The leaf biomass was actually not impacted by carbon dioxide levels, and that's shocking because typically with more carbon dioxide, more carboxylation happens and plants get bigger, but that wasn't really the case here. Rather, what happened is a high water use efficiency line increases root to shoot ratio, and the low water use efficiency line decreases root to shoot ratio. And to be honest, I'm still trying to piece together why this may be. But moving forward, I'm going to be collecting information on seed biomass, seed carbon to nitrogen ratio, and overall seed protein, and seeing if perhaps the carbon dioxide levels had a significant influence there. And And why should you care? So many studies have shown that plant nutrition and seed composition are negatively impacted by elevated atmospheric carbon dioxide levels. And bad seeds aren't just an issue that farmers have to deal with because we all need to eat. And disruptions to agricultural production due to climate change aren't stopping anytime soon. So we have all become so deeply reliant on these agricultural systems, and we need to understand how they're going to change. Because let's be honest, how many of us grow all of our own food? So thank you to everybody that helped me. Dave was an amazing PI. His postdoc, Caillou, did some wonderful work helping me understand my data. Thank you, Faye, for being an amazing human being. And this entire team at the Arnold Arboretum has been so welcoming, and I really feel like there's a community here, and I feel very lucky to have been a part of it. So thank you.
Okay, next up we have Fiona, and she's been working this summer at BU with Pam and Lucy.
Hi, I'm Fiona, and I'm excited to talk about how we are changing our landscapes from the ground up, literally from the ground, because I'm going to be talking about soil nitrogen. And there's a picture of me at the Arboretum doing some field work. So when we talk about nitrogen in the ground, we actually want to start with nitrogen deposition that occurs from the atmosphere to the ground. And that happens through nitrogen ions being dissolved in dust, rain, and snow and falling to the ground. And then they end up sometimes in forests and in the soil of forests. And in urban areas, such as where we are right now, nitrogen, ammonium fertilizer, and nitrogen oxide from fossil fuel emissions also increase nitrogen deposition. And when the nitrogen reaches the forests, plants want to use that nitrogen because, as Mary was explaining, it's a very essential nutrient for life. But plants can't use that nitrogen unless it's in inorganic form. So when the nitrogen reaches the ground, it needs to be transformed from organic nitrogen to the inorganic nitrogen. And in this process, some carbon and some nitrogen are also a byproduct and can be stored in soils. And another way this happens is when plants and animals die, their organic nitrogen can be transformed to inorganic nitrogen and returned to the ecosystem they came from. And so this process of inorganic nitrogen coming from organic is called mineralization. And microbes perform this for plants all of the time. And here's the more complicated version, but you can think of it as the production of ammonium and nitrate. And then here are different ways that nitrate will leave the system eventually. So from what we know about nitrogen deposition, I hypothesized that an increased nitrogen deposition, especially in urban areas and on actually forest edges too, because forest edges are also usually vulnerable to fossil fuel emissions, for example. I expected that rates of nitrogen mineralization due to increased nitrogen being available, those rates would increase because more ammonium and nitrate could be produced, and then more carbon and would be stored in the soil because trees are growing more after having more available nitrogen to take up. And then they would also be decreased levels of nitrogen because it would be used more to make more trees and plants. And so then I also hypothesized that there would be an increased carbon to nitrogen ratio due to this increased carbon and decreased nitrogen. And so we were testing our hypothesis using this urban to rural gradient and these 8 sites from the Arnold Arboretum to Harvard forests. And at each of the sites, we had this edge to interior transect, and so that way we could think about the ways that the forest edge effect was impacting soil nitrogen. And so we had these different points that we were measuring from, and we used buried bags method, which measures net mineralization with 2 different measurements, a final nitrogen amount and an initial amount, and then you can get your net. And we were also measuring soil carbon and nitrogen, those pools that I had up earlier. And so to do that, we were measuring them on autoanalyzers. And so it was really interesting. These results are from 2018, in which a very similar study was performed. 2019's results were not ready yet, unfortunately, but we saw some very interesting trends in that in edges, in this area on the edge, so distance from edges on the x-axis, and you can see that it starts at 0 meters from the edge and goes to 90. And on the edge, there was a clear dip in ammonification and mineralization. And then as you went further in the urban sites, it increased significantly for ammonification and nitrification. But we also saw that there was a different trend happening for nitrification. So something that I think might be going on is that ammonification may be the more dominant trend, and so maybe more ammonium is being more regularly produced rather than nitrate. I'm not really sure why that would be happening, but it would be an interesting thing to look into. And then for the pools of soil carbon and nitrogen that we were measuring, we noticed these are just urban sites, and we noticed that they also seem to be lower amounts of carbon on the edge and nitrogen on the edge, which is interesting because you would think that you would normally see increased biomass on the edge due to there being more space to grow on an edge versus on an interior of a forest. But we actually saw less carbon. So again, I'm not really sure exactly what that means, but it would certainly be interesting to keep on looking into that. And we also saw no change in CN ratio, which is interesting because as you go from the edge to the interior, you might expect there to be a difference in what is being decomposed, but actually there seemed to be not much of a difference. And so I also wanted to look specifically at the Arnold Arboretum because everyone here is very interested in that. And so it was amazing because even though in the general urban sites, when I kind of averaged them together, I noticed this downward trend on the edge in the Arnold Arboretum itself, and here's where our site was in the Arboretum. I saw that there was increased carbon and increased nitrogen on the edge, which is strange, and I am not really sure why it would be different from the averaged urban sites. And we also saw a different trend in carbon to nitrogen ratio. And one of the reasons why it could be different is just due to there being a different ecosystem here than the other urban sites. And the Arnold Arboretum is a particularly unique place. So maybe that's why it's different. But my conclusions were that the urbanization effect that I mentioned earlier seems to intensify rates of net nitrogen mineralization, producing more ammonium particularly, but does not change soil carbon to nitrogen ratios. And then the forest edge effect that I mentioned actually decreases rates of net nitrogen mineralization and reduces amounts of soil carbon and nitrogen generally. And then the Arnold Arboretum in particular seems to have higher soil carbon and higher soil nitrogen and a lower carbon to nitrogen ratio. And so these seem to be kind of conflicting. The urbanization effect and the forest edge effect seem to have conflicting effects. And so it'll be particularly interesting to keep on looking at this and figure out maybe if there's one is more dominant than the other, or how they're both interacting, especially as our forests become more urbanized and more fragmented. So thank you to everyone who helped me. Thank you to Pam and Lucy and everyone at the Templar Lab and the Hutira Lab and the Arnold Arboretum. And yeah, thank you.
All right. Next up, we have Danny, and he's been working this summer in the Friedman Lab.
Hi everyone, my name is Danny. As Space said, I'm working in the Friedman Lab, and so my project considers, it looks at how leaves of trees of the walnut family change throughout the growing season. So before I start talking about my research, I wanted to delve a little bit into the framework through which my research is being conducted. And this is the idea of the leaf economic spectrum, which posits that by looking at these 6 characteristics of a leaf, namely its lifespan, leaf mass per area, photosynthetic capacity per unit mass, you can better understand or even predict its role in a plant's role in an ecosystem, and you can also understand how a plant invests essentially into its leaves through carbon. And so I wanted to draw an example. I like to think of the leaf economic spectrum as being on this sort of continuum from tortoise to hare. On the tortoise side, we have a plant like the monkey puzzle, which is an amazing name, and has 21, and its needles can be expected to live for 21 years, whereas this plant, the ocotillo, only puts out leaves after rain, and those leaves will fall out after just 2 weeks. So there's a very large difference between leaf composition, and this can imply very different things about the ecosystem that they're adapted for. And so an ecologist would essentially take these six characteristics, boil it down to a number, and plot it on a graph essentially like this, that or multiple graphs like this, that will essentially have things that will do more, that will be more of like a hair type strategy versus more of like a tortoise-like strategy. And so, while I have this, this is my little graph here, they actually deal with things that have thousands of leaves and look a lot more scary. So, so while they do that, they generalize these plants by looking at one leaf at one point in the growing, looking at a couple leaves at one point in the growing season, at one point on the growing axis. But as we can tell from one of my study species, Caryophyta right here, they produce very many different types of leaves, and leaves change at different points throughout the season, and they put out different numbers of leaves based off of the species. So as you can see, this is a Caryophyta leaf with just 5 leaflets, whereas this is one with 3 leaflets. And the bud over here produces these specialized scale leaves, which are also leaves, but they are very, very different in terms of composition and function. And so this is one of my study species. No 2 leaves are the same. That's the big point. Sorry. And so while this is one of my study species, this is my other one, Juglans nigra, and as you can see, it puts out a significantly different number of leaves. And so why are they putting out these different number of leaves, and how are they putting out these leaves? And so in order to do that, we have to go into the different ontogenies, which are essentially the strategies that they're using to put out these leaves. And so we have bud bursts in April, and as we progress, both Caryophyta and Juglans nigra are putting out their leaves. But once we get to June, all of the preformed leaves that were created in the previous growing season are put out. And Caryophyta no longer puts out leaves, but Juglans nigra continues to put out leaves all the way into September in the fall, when they stop neoforming leaves as well. And so you would expect that a leaf put out in April would have to last 6 months, whereas one put out in September might only have to last 1 month. So a leaf that is put out in April will have to deal with frost damage, whereas one in July wouldn't, and will have to last for a significantly different period of time, and will have a different lifespan. So my hypothesis in looking at the study is that Caryophyta, which is my more tortoise-style plant tree, will have a higher structural investment because they're expecting a season-long leaf longevity. They're going to have a lower photosynthetic productivity overall, whereas Juglans nigra, my more hair-style plant, is going to have a lower structural investment, a lower leaf longevity, and a higher photosynthetic productivity. So in order to do this, I've been going out to the grounds on the Arnold Arboretum, looking at mature trees, as well as going to Common Garden and looking at juvenile plants of these. This is me measuring my 10,000th minicam measurement, which is a big moment for me. And we're using a minicam, which measures the photosynthetic yield of photosystem 2, which is correlated to assimilation. Also measuring the length of each leaf to get an approximate area, which to convert it with the correlation curve that I made. And so this is essentially what we found. And this is a pretty complicated graph, so I'm going to walk you through it. We have 4 time points along the x-axis, April, May, June, July, since bud break, and eventually this will go all the way to September or October. And on the y-axis, we have the leaf position on the growing axis, where leaf number 1 will be the first leaf, leaf number 2 will be the second leaf, and it'll keep going all the way until like leaf 15 or so here, which will be the newest leaf, and leaf 1 will be the lowest leaf. And so the size of the dot corresponds to leaf area. The amount green it is essentially corresponds to how much photosynthesis it does. And this frequency right here isn't very easy to see here, but that depends on how many plants actually have that leaf on that position in the growing axis. So what we're seeing here is as spring kind of progresses, in May we're seeing these scale leaves from the previous season, which would be down here, come out and grow out, do a little bit of photosynthesis before they fall off. But once we reach June, all the leaves that Caryophyta puts out, all the foliage leaves that Caryophyta puts out are out. And they're not progressing, they're not putting out any more throughout the season, and they all have a somewhat mostly constant photosynthetic yield throughout each time point. And so while Caryophyta does this, Juglans nigra has a very different looking graph, essentially. And so we can see that there's that while the leaves are still coming into shape, Juglans nigra doesn't stop putting out leaves and hasn't stopped putting out leaves yet. We can see that there's a difference in the photosynthetic yield between each time point. And the most interesting thing that I found is if you're to track if you're to track the first leaf from April to May to June to July, you see that while most have the first leaf in April and May, none of them had it in June, and in July, none of them have the second leaf, and very few have the third leaf. So what we're seeing is instead of these leaves lasting 6 months at the beginning of the growing season, the ones put out at the beginning of the growing season lasting 6 months, we're seeing them fall off early. And why might they be doing this? They might be doing this in order to reallocate resources for other leaves, and which might allow them to do photosynthesis better throughout the growing season. And so using the leaf economic spectrum data from the study I showed you earlier, I plotted Juglenseiger and Parda ovata with their single leaf leaf economic spectrum measurements, and so this is where an ecologist would put it. And while these are, while what I found is in line with the ecological perspective, they're not necessarily understanding the heterogeneity that I talked about earlier through the different leaf types and the different points in the growing season might complicate this a little bit more. So by really understanding and analyzing the leaf mass per area and nutrient composition of the leaf, as we would do in the future, we can understand leaf economic spectrum a lot better. Thank you very much. I want to thank my grad student mentor, amazing grad student mentor, Crystal, for helping me out so much with this project, Professor Friedman, Dr. Fay, the DaRin Butz Foundation, and the Arnold Arboretum and Harvard University. Thank you so much.
Cool. No? Or did someone raise a hand? No. Okay. All right. Next up, we have Emma, and Emma's been working in the house.
Do you want the thing? It's over here.
Okay, hi, I'm Emma. I worked in the Hopkins lab this summer studying speciation in plants, and today I'm going to talk to you about my project, which was an analysis of the levels of reproductive isolation in phlox. So taking a step back, speciation is the process of forming a new species, and a really important thing to understand about speciation is that it's a continuous process. So if we look at it over an evolutionary timescale, it goes through multiple different stages. And so the way that it works is you have this common ancestor, which is one species, and two populations of that species diverge. And they can diverge for a lot of different reasons. So maybe they become geographically isolated, and so now they're evolving and adapting to different environments. Or maybe they still live in the same area, but they have different pollinators, and so that's causing them to evolve and adapt differently. But once they do diverge, they're in this stage of early divergence, where there's still reproduction between the two populations, there's still gene flow. But as time goes on this evolutionary timescale, and as these two populations continue to evolve and adapt, they go through this stage of intermediate divergence, where reproduction continues to decrease, and gene flow between the populations continues to decrease, until ultimately you're in this stage of late divergence, where now these two populations are completely reproductively isolated. And so essentially the hypothesis is that these less diverged species will have more reproduction between them, and these more diverged species will have less reproduction between them. And so if we think about how plants reproduce, a lot of things have to happen. So if we look at this flower bud as an example, and we don't think about self-pollination, so not really focusing on those pollen grains within the flower, pollen from a different individual has to come to this flower bud, it has to bind to that stigma, that female reproductive organ, then has to germinate, so grow that pollen tube. That pollen tube then has to travel down the style to the ovary where fertilization has to happen, right? So you need a fruit to swell to get seeds. And so at any one of these stages, plants can reject this pollen, and you can have these barriers to reproduction. And so what I was really focused on was looking at pollen binding and pollen germination, and seeing if and how they act as barriers across different phlox species. And so we can look at these barriers on the confocal microscopes here. So in this top photo, that's a pollen grain that's actually bound to the stigmatic surface, so right about here in the diagram. And in this bottom photo, those are several pollen grains that are bound, and actually one of them has also germinated, so grown this pollen tube that's going to travel down the style. And so what I did is I looked at 5 different species from an area of sympatry in Indiana. And so this is the phylogeny of these species, so you can see that there are varying levels of divergence. Some of them diverged longer before others, and then these are photos of each of the flowers of these species, so you can see they're really diverse phenotypically. And then also the map of the geographical ranges of each of these species, so they're all overlapping across this range. And so what I did is I artificially crossed individuals from different populations across these 5 species. And my first main question in doing that was to just try to understand how compatible are these species. So what are the varying levels of reproduction between them? And so to do that, I quantified the number of pollen grains that bound to each stigma and the number of pollen grains that germinated for each cross that I did. And in doing that, I was able to calculate this value of reproductive isolation. And so that's what you see here in this heat map. So on the x-axis is each of these species as the father. On the y-axis is each of these species as the mother. Those boxes with slashes through them were ones that we didn't have data for. But this value of reproductive isolation ranges from -1 to 1. So 1 being that dark blue color means complete isolation. 0 being that yellow color means that these two species actually reproduce with the same success that they do within their own species. And -1 being that dark red color means that these two species actually reproduce better with each other than they do with themselves. It is worth noting that there are 3 species crosses here that are done within the same species. And so for those, we're looking at different populations to get this value. But what I want to draw your attention to is when we think about speciation, and the way that I described it to you, we think about 2 populations that are diverging over time. And as they diverge, they lose this ability to reproduce. So we'd expect these really distantly related species to have high values of isolation. And so if we look at 2 distantly related species here, so if we look at Phlox divorcata and Phlox bifida, and we look at a cross between them, we do see this high value of reproductive isolation. These 2 species can't reproduce. But if we look at the same 2 species, but now we reverse who is the mother and who is the father, so we do the reciprocal cross, so now Phlox bifida is the mother and Phlox divorcata is the father, we actually see a very low value of reproductive isolation. So in fact, they can reproduce. And so this is what we call an asymmetry in reproductive isolation, meaning it really matters who the mom is and who the dad is to determine if they can reproduce. And so with that in mind, I then wanted to think about which barriers were dictating this compatibility. So was it pollen binding or pollen germination that seemed to be responsible for this variability in reproduction? And so what you're looking at here on the x-axis are each of the species pairs that I crossed, and they're organized, they're grouped together by reciprocal. And on the y-axis is this value of reproductive isolation. And this is all in terms of pollen binding, so the number of pollen grains that are bound to each stigma. And so you'll still see those asymmetries within these reciprocal crosses, and you do see some isolation values that are high, but there's actually only one species pair, Phlox maculata times Phlox paniculata, that actually has this reproductive isolation value of 1, so no pollen binding. But when you look at pollen germination, you see that a lot more species pairs are still having these really high values of isolation. And so what this tells me is it seems to be pollen germination that's preventing these species from reproducing. This seems to be the barrier that's dictating this compatibility. And in fact, when we look at them together, we do see that on average, pollen germination has a higher value of than pollen binding. And so what can I conclude from what I've told you so far? So first, divergence does not always predict a reproductive isolation. So again, thinking back to that heat map, even those 2 really, really distantly related species can reproduce depending on who the mother is and who the father is. And similarly, reproductive isolation is asymmetrical. So again, the direction of the cross makes a difference in how well they're going to be able to reproduce. And then also, pollen germination seems to be this barrier that's dictating this compatibility. So again, thinking back to that pollen grain that's germinating and going into the style, that seems to be the point at which these species are unable to reproduce. And I just wanted to end on why I think studying speciation is important. So just a fact that you can find on the Arboretum's website is that there are over 2,000 different species in the living collections here. And so if you think about it, each and every one of those species has to undergo this process of speciation over evolutionary time, right? All living things do. And so if we can better understand how and why these new species are forming and what the barriers are that are keeping them apart, we can work to better maintain and also create the biodiversity that we have not only at the Arboretum, but also in the natural world as a whole. And with that, I want to thank the Hopkins Lab, specifically Robin, Austin, and Matt. I want to thank Faye, the DaRin Butz Foundation, my fellow interns, and of course, the Arboretum. Thank you.
Any questions for Emma? All right, thank you very much. Next up, we have Danielle, and she's been working with Beth Briggs this summer.
Yeah, it's on. Okay. Whew. Hi, everyone. My name is Danielle, and this summer I explored morphological trait relationships in the genus Castanea. And in this genus, we have the famous American chestnut. The American chestnut could be found along the Appalachian region, ranging from northern Florida to southern Maine, and west into Tennessee and Ohio. And the chestnut trees grew absolutely huge. They had heights well over 100 feet. and could have diameters of more than 10 feet. But unfortunately, in the year 1904, everything changed when the chestnut blight attacked. Chestnut blight is a fungal pathogen that was accidentally imported to the US from Asia. As a wound pathogen, it enters the tree through an injury in the bark and spreads to the underlying tissue, killing the tree as it advances. Now, to keep in mind that all the species in the genus Castanea are susceptible to chestnut blight, but it hits the American chestnut the hardest. Since the introduction of chestnut blight, many chestnut trees were dying by the 1950s. This may seem bleak, but don't be too sad. Nowadays, chestnut restoration is becoming more attainable through approaches using traditional breeding, biotechnology, and forest management. The American chestnut belongs to the genus Castanea, which is home of the chestnuts and chinkapins. It consists of 8 species and 3 known hybrids. Here I have a picture of 2 different phylogenies from 2 different papers. So you can visualize that there's still conflict going on between the genetic backgrounds in this genus. But one thing we can all agree on are the 4 main groups: the Americans, the Chinese, Japanese, and the European chestnuts. So I'm interested in Castanea because I want to provide more background information about the morphological traits to help fill the literature gap, because in literature there is not enough information looking at the genus as a whole, and so that's where my part comes in. My objective was to analyze the morphological traits and describe variability in Castanea in order to understand what relationships are going on. This helps contribute to evolutionary research, and I think identifying traits and correlations in Castanea will give more insight about how to better protect these species. Data. Okay, so here are some of the traits I've been measuring this summer. This is things relating to leaf size, leaf area, and this is just showing you a nice variability of what I've been looking at in this genus. So this is a lot of stuff to look at. To analyze all this trait information, I use something called Principal Component Analysis, or PCA for short, to understand the correlations between traits and show major axes of variation in my data set. So if we read my data that I've collected this summer, 65% of the data can be explained by the traits found along the x-axis. For example, would be these traits right here relating to leaf size. And each color that I have up here represents a species. If you imagine quadrants, like I've pointed out here, in this quadrant, you'll find leaves with more leaf area, more serrations, and longer length. And in the opposite quadrant, you'll find shorter leaves with less serrations and less leaf area. And so to make this a little less overwhelming, we're going to take a look at just a couple. We have the American chestnut and the Chinese chestnut. And in the PCA, same data as I showed you in the last slide, just highlighting these 2. The American chestnut and Chinese chestnut, you can see they occupy somewhat of the same space, but also 2 different spaces along a PCA. One of the biggest factors is what's influencing the y-axis, which would be the lateral vein angles that I've been measuring. And overall, the American chestnut tended to have bigger vein angles than the Chinese chestnut as I was measuring this summer. Another example from our PCA that I showed you earlier are the chinkapins. There is genetic data that shows that there are 3 distinct genetic groups going on within the chinkapins, and my morphological trait data helps back that up by showing 3 distinct regions, or 3 regions also being shared by one another, but like the Ozarkensis, if we look at that one, versus Pumila 2, you'll see like Ozarkensis will have bigger leaf area rather than Pumila 2, who is found on the opposite side of that side of the PCA. So it will tend to have less leaf area than Ozarkensis. And here is everyone. This shows this shows that there's nice overlap and trait variation going on within the genus Castanea. In conclusion, there's many trait characteristics that I found within this genus, and this can help resolve current taxonomic disagreements, like how I showed you with the chinkapins. And also, increasing the knowledge surrounding this genus can help improve current conservation efforts for the chestnuts and chinkapins we know and love today. Thank you.
So next up we have Erin, and she's been working with Kat this summer.
Hi everyone, like Faye said, my name is Erin Wright. I'm a member of the Holbrook Labs. And this summer I looked at the relationship between phenology and flowers. So first off, what is phenology? Phenology is the timing of life history events of different organisms. And when you first think about it, you might think about when do beetles emerge or when do geese migrate. But looking at it through the context of plants, we think about when do plants put out their leaves, and maybe when do they open their flowers or go to seed. And as we live in a warming and changing world, climate change is affecting the cues that these plants use to time their life history events. And this is especially expressed through these fall spring events. And a fall spring event is when it warms enough that the plants think it's time to put out their leaves, but then it quickly cools again and drops below freezing, causing intense frost damage, as illustrated by these two images taken by Kat Chamberlain. And these plants, through this damage, are going to have to spend the entire season regrowing that tissue and reinvesting photosynthate into that tissue, setting back a year of growth. And this summer I worked in the system of a common garden, and in these 4 red points you can see you see 4 different latitudes that we took seeds from, and they're all being grown in the same location here at Weld Hill Research Facility in the common garden. And it doesn't quite look like it on this map, but these 4 points occupy a very wide range of more than 500 kilometers or 300 miles. And there are 7 species that I looked at this summer. In the bottom right-hand corner, you see Myrica Gale, which is the longer red catkin is the male flower that at that time was putting out pollen. And in the bottom, and the smaller green one is the unripe fruit. In the middle, you see the flowers of Sambucus racemosa and the flowers of Spirea alba in the top right. Now, before we dive any deeper, there are 2 key points I want you to take away from this presentation today. And the first is that phenology is an important tool that we can use to study climate change. And that secondly, by using a common garden, we can begin to ascertain the population variation within these species. And there are 2 major questions that I hoped to begin to answer this summer. The first is, how is climate change affecting the timing of life history events of plants? And to dive a little bit deeper into that question, how do floral and fruit morphological traits affect the timing of these phenophases? And there are 3 major hypotheses that I hope to test. The first is that the later that bud burst occurs, the shorter the amount of time that the flowers will be open. And this is because that gives the plant less time to have these flowers open and then to go to seed. And the higher the latitude of origin, the shorter the period of time that the flowers will be open. And this is due to, at higher latitudes, there's a shorter growing season, and again, shortening the amount of time that these flowers have to reproduce. And the higher the latitude of origin, the later the flowers are open, and that's because we expected that they would open their leaves earlier and therefore push back the time of flowering. And to test these hypotheses, we use phenological observations, basically just taking down the day of the year that these certain events occurred out in the common garden, and then measuring the floral traits of these flowers, such as their size and their dry mass. So there was really no correlation with these floral traits and either latitude of origin or the phenophases. But what we saw was relationships with growth. So firstly, on the x-axis, we see here we have the day of bud burst, and on the y-axis, we have the seasonal growth measured in centimeters of height difference. And to start off with, we see that with Sambucus racemosa, as the day of bud burst got pushed back, the seasonal growth decreased, which is what we would expect to see, because later bud burst means less time to grow, and therefore less growth. But then we look at Viburnum casinoides and Myrica Gale. As their bud burst time pushed back, they actually grew more, which is something really interesting and kind of unexpected. And then secondly, we're looking at Sambucus racemosa again, but on the x-axis this time we have provenance latitude, and provenance is just the origin where these seeds came from. And on the y-axis again, we have seasonal growth. And as provenance latitude increased, the seasonal growth for Sambucus decreased. But for Emelanthier, it increased as the latitude of origin increased. And just to draw some conclusions from this, there is no correlation between flower and fruit morphological traits and the phenophases and their latitude of origin. And then just to tie it all back together, phenology and climate change can be explored using the Common Garden. And from these results, we can kind of conclude, they suggest that there is significant genotypic variation left for them to respond to the environmental cues around them rather than what has been programmed into their genomes genetically. And I would like to thank Faye and the DaRin Butz internship program and the Arnold Arboretum, as well as Kat, Professor Holbrook, and Anju. Thank you.
Any question for Erin?
Do you think the interesting ones that grew better when they leaked out later, I think that was the pattern? Was it because they were avoiding some injury that associated with leaking earlier? Or?
Not necessarily. It could just be the point that they are in their life. Maybe they're growing more. So maybe just like a species variation. It could be that they're avoiding certain things, but again, we didn't really see that correlation with like latitude of origin and like any of their like other phenophases and stuff. So that really doesn't seem like to be the case. Yeah.
So those were all the topics. So we have time for any more questions if you would like to ask them or not. And if you don't, we're all done. So thank you all for giving wonderful talks.
Well, 10 weeks goes by very fast, doesn't it? Thanks to all of you, great job, and thanks to everyone here who made these projects possible. It's not just in the labs, it's the plants that are out there on the grounds that have to be healthy to be studied. It's all of the sort of mentoring that's been going on. I just think it's been great to see 10 people come here and really engage with the institution. And a huge high five for Faye. She's the one who's gone after the funding that makes this possible from the DaRin Butz. She's had an incredible mentoring relationship with everybody here, and simply put, this wouldn't exist without her vision for creating this program at the Arboretum. So thank you, Faye.
And of course, don't forget that we have a barbecue at noon.
Yay!